So I'm delighted to be here, Minister, ladies and gentlemen. Thank you, and Fred, for inviting me to speak today. I'm going to talk about one of the smallest life forms in the world and one of the oldest. So Ben actually talked about something that we use in microscopes, so it was lovely. I'm not going to explain all of that stuff, but it's exactly what we've been using as point spread functions, deconvolution, and other things that you mentioned. So I'm an Emeritus Professor. That means I'm being acknowledged by UTS, where I used to work, as contributing in leadership, mentorship and research. I still belong to that community. I have an office there, but I'm actually, my employment and my business is as a Senior Associate for Outside Opinion. We give expert advice and support to researchers, academic researchers, to support them doing leadership, strategy, grants and fellowship writing, and also just to be the best they can. And it's been extremely rewarding to do that, just to find out what they really want and do it their way. Okay, so how are bacteria discovered? What I'm going to talk about is some very basic stuff about bacteria. How do we know they're there? How did we find them? And some of the technologies that we've used to be able to, so that they're not so secret anymore, although there's always more to discover, of course, in research. So the first person to find or see bacteria was a Dutch scientist in the 1600s. He was called the father of microbiology, Antony van Leeuwenhoek, and he made a simple magnifying lens of 260 times magnification. He was not a scientist, he was curious, and I think that's the key thing, is that if you're going to, doing research is purely about an insatiable curiosity that you have. And he called these microbes animalcules, and these were not just bacteria, and I think that's the pointer there. Yeah, this is on your right-hand side. So these were not just bacteria, but mainly single-celled organisms, including bacteria, and these are his actual drawings. So no one had ever seen this before. No one ever thought that there would be such things in the world, ever. Okay, and then Koch's postulate. So Robert Koch was a very famous microbiologist in the 1800s, and he was from Germany, and he was the first to prove that a microorganism could actually cause infectious disease. And he showed that if you take a healthy animal and look at take blood from it and put it on an agar plate where bacteria feed on and grow on, that nothing grows. But if you take an infected, or what he thought was an infected diseased animal, and you take their red blood cells and put them on this agar plate, you see this pathogen that we call bacteria that causes infection grow. Then if you take that bacteria from the plate and inject it into an otherwise healthy animal, you then take their blood and you find that they have the same organism again. It seems very simple, but at the time people didn't believe you could do it. I mean, science history is full of things we don't think are possible. We still do it, but for good reason. We can't just believe in everything. So he also discovered that bacteria cause tuberculosis, anthrax and cholera, and he won the Nobel Prize for Medicine in 1905. And partly he used new tools here. Before that, we didn't know how to grow bacteria in the lab, just grow them onto agar. We didn't have dyes. We ended up using dyes to show that bacteria have special envelopes on their cells, that certain dyes turn a certain colour. And no other organism does this. So you could tell that they're actually bacteria. So that's very basic. And then microscopy, of course, came along, and it was very simple light microscopy, but not a lens. It was actually a proper microscope. So that helped enormously. And also we arrived in a bacterial world. This is not necessarily just a human world. It's really important to understand that these guys have been there for a long time, because we rely on them, we depend on them. And they're really important, obviously for our health, but also for a whole range of reasons. Now, bacteria are 4 billion years old. Modern humans are 200,000 years old. So they've seen a lot. They've seen a lot of the molecules that we provide as antibiotics. It's one of the reasons why they're resistant, because they already have, but they already were. But we know that they're this old, mainly because of stromatolites in Shark Bay in WA. When I was at Harvard, I heard Stephen Jay Gould talk about these in his lectures. He's a very famous evolutionary biologist. So what happens is these microbial mats formed between the layers of these sedimentary rocks or sediments. And so they built up over time. And if you look inside those sediments, you can see evidence of what's called a cyanobacterium. And a cyanobacterium actually photosynthesizes. So it's just a bacterium that actually is able to do what plants do, that is take glucose, water and carbon dioxide, light, carbon dioxide and water, and produce glucose and oxygen. So these guys, people say, gave the kiss of life by providing oxygen to the atmosphere. Of course, this is what we need, but many, many organisms need that. They're very, very, very small. So I love these pictures that show on a household pin on the left-hand side is purple, and the bacteria, these little orange things. They're about 2 microns long. That's showing you a larger version of the agar plate with the hand holding it. So what we call these little spots are single colonies of bacteria. And the way we do experiments is just to take one of those single colonies and inoculate it in a sterile way into a broth, grow the cells, and do experiments with them. 1,000 times over on the right-hand side is actually just a light microscope. So you just see a 2D version of the cells, and that's just a rod-shaped cell. What's been around for a long time too is electron microscopes, and they measure density basically. So you put your bacteria, you embed them in a gel or a wax, you slice it up like a really precise thin guillotine, and then you look at the density. And what you can see is the envelope there, and if you're close enough to the front, you can see that it's undergoing a division. This is again a rod-shaped cell, and the little blobs in the middle that are a bit lighter is actually the DNA. So again, this is the sort of information we could get. It's not a lot, and a lot of the work came later when we could do DNA sequencing, molecular biology, and better microscopes. So what I wanted to say is, while the average bacteria is about 2 microns long, the largest bacteria was discovered only 3 years ago. So in 2022, a Science article reported a organism, a bacterium that could grow up to 2 centimetres long. And what it does, it divides very differently. This is it on the left-hand side. The scale is 0.5 millimetres. So it's bigger than a fruit fly, which is extraordinary. And E. coli is down here at 2 microns. So this is extraordinary. And it does divide in a similar way, but it also has compartments where the DNA is. It has many copies of the DNA, which is very unlike bacteria. They tend to just have 1 chromosome. So we will continue to discover these new ones, which just keep informing us about life. And life on other planets, perhaps, we'll find all sorts of different types. They exist in all sorts of shapes, and these have been coloured a bit. They don't all look, that artistic, but they do look very cute. They're often rods, and there's some spirals down there in the bottom. Sometimes they're in chains of balls. Sometimes they like to keep as diplococci, which are 2 cells together. We still don't really know. I'm fascinated by why they don't just separate and live as single cells, but they just divide and separate properly, but they hang on to each other. There's also tetrads, which you can see, the orange ones. These ones up here, that's how they like to be. And some of the infections that we have, they're the type that are in us. And the reasons they are these shapes is not totally known. But we do know something about their shapes and why they need to be that shape, and I can talk about that in a minute. The other thing that we didn't really, well, I guess the ecologists, the microbial ecologists knew this, but even that separation of disciplines from microbiologists that are looking at molecular things in the lab, and then ecologists looking at outside in the water pipes and in the seas, they knew that these organisms formed a sort of a slime. So if your soap dish hasn't been rinsed for a while, there's biofilms everywhere, right? They're in our mouth. Everywhere are biofilms. So bacteria tend not to be on their own. They're in mixed species biofilms. And this is very schematic, but basically they attach to a surface, they build a slime, and this slime then, they build this house that they live in. Now this protects them from being eaten by single-celled organisms called protists that graze on bacteria and just eat them. So they don't like this stuff, and we're just trying to work out why. But they don't like to eat them in the biofilms. Biofilms are also resistant to antibiotics, very resistant to antibiotics, and to our immune system, and so it's a defence system. Okay, and one of the things, if you've known someone who's had a hip replacement or any sort of infection where they take antibiotics for 6 weeks, then they stop, and as soon as they stop, a few weeks later the infection comes back again. This is because the biofilms are there. It kills the free-living bacteria, but not the biofilm. So it's a bit of an insidious thing, and eventually, you know, people usually get over these things, but it's really horrible when you get a biofilm. So that's what it looks like under a microscope with this slime, and you see some free rod-shaped cells there. Okay, so the other thing is that they're everywhere. They've been around for 4 billion years, so you'll see them everywhere. So everywhere you look, they are in the air. They also form spores, some bacteria, so they'll be airborne. In the water, there's plenty of them in the marine and freshwater environments. At the park, in the sandpit, which is not bad. It's probably good to get a few exposure, well, a lot of exposure. In your dessert. So, well, they're in cheese, they're in yogurt, and in the desert. In the Arctic, you can find them between ice layers. They live in high pH, low pH, high salt, low salt, and on your skin, and of course inside human beings. So how many bacteria in and on our bodies? About trillions. So there are 10 times more bacteria in us, more bacterial cells in us than there are human cells, which many of you would have heard. And our poo contains almost half mass of bacteria. So if you take the water away, what we excrete is mainly bacteria and other waste products. So this is why people are doing poo or fecal microbiomes to see how healthy you are. And it does tell you a lot about what's happening in your gut. And that's why it's important that you eat the right foods and take a look at what's happening. And you can actually do a poo microbiome in many places in the world, including in Queensland. So where are they on our bodies? There are some places you don't want them to be, for example, the brain and the blood, but they are in our gut, mouth, skin, nose, anything that's accessible. And most of them are doing really good things to protect us, and they're critical for our health. If we didn't have bacteria in our gut, we wouldn't be able to digest a lot of the food that we have, and we would also have infections in our gut. If we didn't have them on our skin, so we have staph on our skin and various other bacterial organisms, if we didn't have those, we would have fungal infections and all sorts of things. So they're actually doing us good. And remember, they were here when we came along, so they just went, okay, great, let's move here. But most of them do good. If they didn't do good, they wouldn't be around, so that wouldn't be, sensible. And you would have heard of the human microbiome. So there's a lot, and again, Ben mentioned sequencing and how we can do it so much more quickly, so much more accurately. It's extraordinary in my lifetime as a scientist what we can do. We used to do like 2,000 base pairs, something like 500 base pairs a day. You used to pour a gel, used to take 2 hours, rinse the glass plates, pour the gel, use radioactivity. Now it's just billions, trillions of bases, it's just ridiculous. It's wonderful. Very few bacteria cause disease, and this is the really important thing. So people think of bacteria and think, yow, yuck. Well, most of them don't cause disease, and we couldn't live without them. So this is estimated to be 1%, but that's probably a well overestimation, because we're going to find a lot more outside in the environment in the future than we're going to find in the body. So they're from the air, like in tuberculosis and meningitis. And as you know, tuberculosis can be caught by coughing. From insects and Lyme disease. In fact, 60% of infections in humans, bacterial infections, are due to transmission from animals. So it's called zoonoses, so it's an important concept. From direct contact, so this is anthrax and leprosy. From food and water, cholera and typhoid. And from our skin. So this is tuberculosis, or Mycobacterium tuberculosis on the left. Staph aureus loves to live in little round cells, and this is not a very nice wound. So the reason why people often ask me, why do you have an infection in the wound of staph when staph is meant to be good for you on your skin? And infections happen sometimes when you ingest things that have bugs in them, but the other reason why you get infections is when bacteria are in places where they're not meant to be, right? So when you cut yourself, you go gardening, it's really important, particularly with antibiotic resistance around, to cover yourself and wear gloves. Or when you cut yourself, put something on it, wash it straight away. It's quite serious, because you could end up with a resistant infection. And soil is a major place for bugs that are not very nice for our wounds. So we can't live without them. I've already said quite a bit of this, but it's estimated they provide half the oxygen we breathe, 80% of the nitrogen we use, which makes protein. They're needed for our digestion and protection, and of course in other animals and living organisms. They eat oil, They're very good at doing that. Again, not all of them do this. Just recently it was shown, well, it was about 10 years ago in Japan that they found bacteria that could digest the plastic that's in PET bottles. Hopefully they'll find more that can digest other plastics, but it's incredible what they can do. They're also biological factories, so they're used in industry a lot for producing things like vitamins and detergents. They're also used as probiotics, which you would have heard, and these are whole organisms which are either dead or alive that you can take for gut health, and of course in food. So how do they multiply? Well, we still don't know exactly how they multiply, even though it's a very simple process. It's just a splitting off of the cell. But you can imagine bacterial cells have to do that by using lots of different proteins and a machinery of some sort. And they can't just do it willy-nilly. They have to do it at a certain time, at a certain place. Otherwise, you don't get one chromosome in each cell. Okay, they only have one chromosome. They double that when they replicate the DNA, and then it has to split. And so if they don't get the right amount of DNA or the right copy, then both cells die. So it's really important they do this properly, and I became very interested in how they regulate it. So these are just electron microscope images of these cells dividing, and they all divide slightly differently, but they use very much the same proteins to do that. And okay, so what I did was identify the first division genes, and this was in the 80s. And one of those proteins, and it doesn't matter what it's called, FTSZ and Div1B. So it turns out over the years FTSZ has now become an antibiotic target, because if you stop division you can kill cells, and of course infection is multiplication. They have to multiply to cause an infection. So this has been interesting. It's taken years to get to this point, but little did I know at that point that this would become a major protein of interest. And one of the questions we were asking was, if this FTSZ protein, we knew that it marks the division site. We showed by microscopy that it's the first protein to go to the division site. We knew this through a microscopy I'll talk about in a minute, but basically it establishes where division is going to occur. So if you want to know where division occurs, you have to work out how this ring knows where to go at the right time. So the question we asked was, how does it find the right place at the right time? And what we found through many, many years was that the initiation of replication, so the very beginning of replication, was responsible for saying this is where you have to form a ring. And there's some proteins that we've identified that are involved in this process. And again, it's a good drug target because it will affect two pathways instead of one. And years later, only because of DNA sequencing, but also X-ray crystallography, which shows the shape of the proteins, and also being able to have the computing ability to look at many, many sequences of different species of this FTSZ protein and other, including our own genomic sequence. And when we do that, what we find is that bacterial FTSZ is very similar to tubulin, which is in us. And major role of tubulin is to form this mitotic spindle that you can see in the middle. It separates the chromosomes like a contractile cytoskeletal sort of element. And so the chromosomes are in blue, and the tubulin is the spindle, which is in green. Now what you can see on the left-hand side is the tubulin dimer. It forms two slightly different molecules that associate together. On the right-hand side is the FTSZ protein itself. And this was discovered the shapes were identified or determined through a lab in Cambridge. And what you can see is that although the sequence of amino acids in these proteins is quite different, but what they could show was that the proteins fold up and form a very, very similar structure. So you don't have to be a scientist at all to see that they're very similar. So this was astounding. We never thought this, so this is why it was in Nature, because tubulin has a relative in bacteria. And we always said, in textbooks it always says bacteria are different because they don't have cytoskeletal elements. They don't have tubulin, they don't have actin, but there's also an actin homologue in bacteria. And it's taken so long because we hadn't been able to localise these proteins and show what they do in bacterial cells, and then also have the ability to compare all these sequences and use the algorithms to identify that. FTSZ is even present in plants. So many years again later, a guy in Melbourne actually identified that FTSZ is present in chloroplasts. chloroplasts do this photosynthetic job in plant cells, and the interesting thing there is that the arrival of chloroplasts and mitochondria is thought to be from originally a cell more like ours actually engulfing a bacterium. So it was a wonderful thing for evolutionary biologists to find this. So seeing inside bacteria is actually really difficult because they're tiny. And again, we didn't have those big ones. We just had these tiny little 2-micron cells. So when I was at Harvard, we decided that we would see, that my supervisor then just said, maybe we should start to use what's called fluorescence to look at various proteins. And we just thought he was mad because, you're not going to see anything because everybody says bacteria are just an amorphous bag of enzymes and DNA. But this turned out to be a revolution. And what you can see here is if you tag an antibody that recognises this protein with a fluorescent marker, or you use green fluorescent protein, and there's lots of different fluorescent proteins from animals such as jellyfish in the ocean, and we can use those using recombinant DNA technology to tag a protein. And so what you see here is just a 2-dimensional image of FTSZ, which is labelled with a fluorescent marker. It sounds silly, but we never thought about where things happen inside bacterial cells. We know division happens in the middle, but I don't know why, but we were all surprised that proteins actually go there because we just had no concept of how it could occur. And it turns out the bacterial cells are highly organised in terms of the proteins in time and space. So they go to certain places at certain times. If they don't do this, if you muck that up using techniques, the process doesn't happen. And so this has been a real revolution because with microscopy getting better and better, we're now able to, and I'm not sure how this is going to work. Yeah, so now using structural illumination microscopy, you can now put bacteria on a side so you can actually see through the ring. And this was, again, a really exciting thing. And we went to a conference and everyone's going, oh my God, you can see it. And it just seems so obvious now. But at the time, so hard to do because they are so small. But really clever. Well, there's been a Nobel Prize for this type of microscopy as well. And it gets better at being able to see more and more inside tiny little things. Okay, the other thing that can happen is that if you use a fluorescent protein, you can actually use it to tag to the protein. You can look at live cells in time. So again, if you look at this, you can see that there's rod-shaped cells and there's things moving back and forth in a sort of a line. Now, we knew from other work in America that these proteins were responsible or partly responsible for determining where division occurs. So and what someone showed was that they actually move back and forth all the time, and they're inhibitors of division. And so the time concentration of these proteins were at the poles. And so what that meant was they stopped division happening anywhere else but the middle. So this, again, was like they'd done all the biochemistry on these proteins, but they didn't know that they did this, and that explained how they worked. And one last story is about how we can now look at infections with great resolution. And how we can look at cell division and how cell shape changes and how we can understand why they change shape and how we might be able to help with stopping those infections or at least treating them. So they're called urinary tract infections. They're in your bladder. Many people have them. There's about a third of the population has them. They can cause all sorts of problems. They can also happen many, many times in your life. They also, main thing is they're antibiotic resistant, many of them, and E. coli is one of the worst ones. They happen in hospitals. If you have a catheter for more than 3 days, you have a really big chance of getting an infection in your bladder. Yep. Sorry, but you need to get out of hospital quickly and get that catheter out quickly if you can. Bacteria invade the bladder cells, so they go into the bladder cells and they change their shape. So just what I wanted to show you first was the bacteria are in purple. The bladder cells are in green, and the whitish sort of stuff is DNA. So you can see that they've just gone inside as short cells. They then just multiply up. And then what happens is you can see in a movie a bladder cell, and the green sometimes it's the bacteria, and it just overtakes that. Yeah, it happens pretty quickly, but to be able to see that live is just tremendous. And it's very blurry here because you can't always get the good resolution when you're doing live cells, but they form long cells there. And this was published in Nature Communications. These are collaborators of mine at UTS. And what they showed, this is a really beautiful picture of the filaments that they form. So they go inside bladder cells. These are E. coli, and they are short and they just multiply. Then as they want to get out, they form these really long cells, and then the bladder cells just burst and they release these long cells. Then they divide up again and go back in again. It's like a virus, goes in, multiplies, comes out, goes in again, as many. And so the blue is the bacteria here. So they form these long cells, and what they're looking at now is how is division inhibited, how is it regulated? And it happens quite differently with some of the same molecules. But what they want to do is prevent filamentation in the first place, because this is where filamentation appears to avoid the immune response. Yep. Okay, so bacteria are survivors. They're old and they're everywhere. They're essential for life on Earth. They're highly organised at the level of protein localisation, and they can disguise themselves very well, and they can change their shape to suit their needs. And there's so much more to discover. So thank you very much for your attention.